Biomedical engineering translates the behavior of living organisms into the strict, uncompromising language of mathematics. The challenge is that biological tissue doesn't behave like steel or silicon. Living systems are defined by their variability, nonlinearity, and constant adaptability. A standard mechanical system is largely deterministic and predictable, but a human body relies on tight homeostatic feedback loops to constantly regulate variables like body temperature, oxygen saturation, and blood pressure in response to changing environments. The engineer's job is to take those qualitative, shifting physiological behaviors and translate them into quantitative, solvable equations. Extracting reliable data from a living human requires reverse engineering the body as an integrated electromechanical system. This reverse engineering process starts by recognizing the body's strict structural hierarchy, scaling downward from the complete organism to the organ systems, and finally to the cellular level. Isolating and analyzing any one of these biological systems requires the convergence of multiple distinct engineering disciplines. Human movement, cognition, and communication are governed by one specific intersection, the point where cellular biology meets electrical engineering. The ultimate clinical goal here is to capture macroscopic physiological data, like the electrical rhythms of the brain or the heart, using entirely non-invasive external wearable sensors. But to accurately interpret the digital readouts collected by those surface sensors, we first have to strip away the gross anatomy and mathematically model the origin of the signal, the microscopic cellular circuit. Every bioelectric signal in the human body originates at a single microscopic barrier, the selectively permeable cell membrane. This boundary separates sodium and potassium ions using a dense lipid bilayer. Acting as an insulator, it functions like a capacitor. Embedded ion channels act as variable resistors, while the sodium-potassium pump consumes energy to maintain the imbalance, serving as the voltage source. This continuous baseline power creates an electrochemical gradient across the cell. In electrophysiology, this baseline state is called the resting membrane potential, typically hovering between -60 and -90 millivolts. By directly mapping biological anatomy to a standard resistor-capacitor circuit, engineers can apply classic electrical laws to mathematically predict how living cells will behave. That resting potential holds steady until an external stimulus disrupts the membrane's permeability. The variable resistors rapidly open, allowing a massive influx of ionic charge, producing a sharp spike in electrical potential. This action potential operates in sequential phases: depolarization as charge floods in, repolarization as channels close, and hyperpolarization before returning to baseline. This local voltage change instantly triggers adjacent ion channels, ensuring the electrical impulse cascades continuously down the length of the nerve fiber. The action potential is the fundamental binary unit of biological information, an electrical pulse that translates chemical energy directly into measurable data. However, a single action potential generates a voltage far too small to be detected from outside the human body. To produce a clinically viable signal, millions of individual cellular circuits must fire in a highly coordinated sequence. This synchronization is achieved through gap junctions. These specialized biological structures physically wire adjacent cells together, allowing electrical current to flow directly from one cell to the next without interruption. The direction and speed of that current are dictated by tissue anisotropy. The physical alignment of the cells themselves acts as a biological printed circuit board, guiding the electrical wave along specific pathways. Macroscopic bioelectric power depends on the physical synchronization of these tissue-level networks. We observe this synchronized electrical power performing actual mechanical work by looking at the cardiovascular system. The central nervous system and specialized cardiac conduction pathways operate as the body's primary electrical pacing network. As the electrical signal propagates through this network, it triggers the synchronized physical contraction of the heart muscle, or myocardium. This physical contraction generates the pressure gradients required to push blood through the vascular system. Hemodynamics, the study of this blood flow, is essentially a complex, non-Newtonian fluid dynamics problem, governed entirely by the timing of the heart's electromechanical pump. Every physical motion and fluid transport in the human body is downstream of an underlying electrical circuit firing. Understanding the internal circuit is only half the equation. The engineer must figure out how to acquire this data at the surface of the skin. Surface readings like an electrocardiogram or electroencephalogram are simply the spatial summation of those millions of deep tissue action potentials echoing outward. But there is a significant physical obstacle. The human body is packed with muscle, bone, and fat. This dense tissue acts as a massive biological resistor, which severely attenuates the electrical wave as it travels to the surface. By the time the signal reaches the skin, it is incredibly faint. It arrives with an extremely low amplitude, often just a few microvolts, and a low frequency range, generally between 0.05 and 150 Hz. The body perfectly isolates its internal systems, leaving the engineering challenge of capturing a faint whisper of data through a thick wall of biological resistance. To make matters more difficult, that faint signal is competing against a barrage of interference. The surface of the skin is constantly bombarded with external electromagnetic noise and overlapping biological artifacts, like stray muscle twitches. Engineers design circuits with high-gain, extremely low noise amplification to boost the microvolt signal without simultaneously amplifying the surrounding ambient static. Once digitized, the software takes over. Because living bodies are constantly changing, engineers use adaptive time-frequency filtering to mathematically identify and strip away non-stationary artifacts, leaving only pure physiological data. It is a complete pipeline of problem-solving. It begins by modeling a microscopic lipid bilayer as a capacitor, tracking that voltage as it synchronizes across tissue pathways, and finally writing the advanced algorithms needed to clean data at the surface. By analyzing the human body through the strict lens of electrical circuits, mechanics, and control theory, biomedical engineers translate chaotic biological behavior into precise, actionable data that drives modern medicine.